A typical single-cell dataset contains thousands of genes, resulting in thousands of variables or dimensions that need to be handled during analysis. That's why we need powerful algorithms, including principal component analysis, t-stochastic neighbor embedding, or uniform manifold approximation and projection to reduce dimensions and better visualize the data. The powerful GPU, accelerated server, and optimized algorithms on BBrowserX can help you perform these computationally intensive tasks in seconds. Now, let's learn how to use these features on BBrowserX. After uploading your data to BBrowserX, click on Embeddings to see the available dimensionality reduction results. A PCA result applied to the raw data, without batch correction, that contains 50 principal components, or 50 dimensions, is the default option. Other results imported along with your dataset are also listed. You can also import a TSV file. The first column must be the barcodes of the cells in the dataset, the next columns contain the values for the principal components. One major issue with using data from multiple batches is that the technical variations can be significant enough that cells from the same batch tend to cluster together. This can result in cell types being scattered on the plot rather than grouped together as expected. This can be solved by running PCA with batch correction between batches or samples. In the Create New section, select a metadata field in the drop-down menu and click Run. Harmony is the batch correction method applied to remove the influence of batch effect from the PCA embedding. After applying harmony, cells of the same cell type are now closer on the plot, indicating that they are no longer clustered together within samples. This suggests that they are now grouped based on their biological differences rather than technical variations. When you select a PCA embedding, a t-SNE plot with the default perplexity equal to 30 is automatically generated from the PCA embedding and shown on the scatter plot. If the dataset contains more than 200,000 cells, a UMAP will be created. To have better control of the scatter plot, you can click on the t-SNE UMAP button. Here you can select one of the input embeddings for t-SNE or UMAP. With t-SNE, you can change the perplexity, which is a guess about the number of close neighbors of each cell. For UMAP, you need to set the number of neighbors. This step will further reduce the data to 2 dimensions for visualization purposes. Between the two methods, t-SNE moves the high-dimensional graph to a lower dimensional space points by points, and only preserves the local structure. UMAP gives a better balance between local versus global structure, thus overall gives a more accurate presentation of the global structure. As a result, clusters of cells are better separated. You can also import the coordinates via a TSV file. The first column must be the barcodes of the cells in the dataset, the next 2 columns must be the X and Y coordinates. All of the t-SNE and UMAP coordinates are saved. Note that the PCA results with more than 2 dimensions are also listed. When you choose these options, the first 2 features will be selected to generate the 2D scatter plot, since these principal components are already sorted based on how useful they are or how much variance they can capture. Next, the PCA embedding can be used as input for LUVAC clustering, which groups cells with similar expression profiles into clusters. First, select one input embedding, then enter the resolution. The higher the resolution, the higher the number of clusters. The default resolution is 1. The clustering result will be displayed on the scatter plot and saved as a new metadata field. For the public studies on the Talk2 data database, we provide 2 default PCA embeddings, one is applied harmony batch correction and the other is not. When you run t-SNE, UMAP, or LUVAC clustering, select one of the 2 PCA embeddings in the harmony section. We hope you find this video helpful. If you have any questions, contact us anytime via support@biotouring.com.